All right, I think we'll get started. Welcome, everybody. Today, I feel like it's my first day. I will tell you, I am much happier talking to people in person rather than staring at the screen and just rambling on, although I will ramble on. It's much better. And I also can gauge if it's going good or not rather than just talking. So hopefully you guys are all up to speed and watched the videos. I think if they're posted, you can watch them faster and maybe that's better. We should be about halfway through chapter 3. I've got intentions of doing chapter 3 and chapter 4, and then 5 and 6 should be pretty smooth sailing tomorrow. I will get an exam posted for after lecture tomorrow. Do you guys have any questions for me right now? Okay. So, and hopefully, I know you've all been here to lab and all of that stuff. Hopefully that's all going smoothly. I will tell you from doing this so many times, I think the summer micro class is awesome. I like just the quick pace. I don't know if you guys will like that or not, but every day we think a little bit about micro, and in the lab, you do something one day and the next day, typically the next day, you get to see some results. And in the regular semester, you don't see it for a whole week. And by the time that next week rolls around, you're like, now, what was this plate? What did I do here? What really happened? You remember it a lot better when we're doing it like this, even though you're like, holy cow, we're doing all of this in 16 days. Are you kidding me? We'll get through here. It's going to be just fine. All right. So if you do have questions about stuff, lab or lecture stuff, let me know. I can definitely help out with some of that stuff. So we got a good chunk of the way through the first handful of chapters, part of chapter 3, and I left you guys off last time, yesterday maybe, or late last week for me. I left you off right here with the start to think about some eukaryotic cells. And there are all sorts of details when we think about comparing prokaryotes to eukaryotes, when we think about, I'd mentioned size and things like that, with prokaryotes being considerably smaller. But we also need to think about what these cells are doing. And it doesn't really matter if we're talking one of these single-celled eukaryotes, one of our own cells in our body, or a bacterial cell. These cells want to live, right? They want to pull in nutrients if they want stuff, right? They pull in nutrients, they metabolize, they break these things down into specific monomers, harnessing energy, and in turn using some of these monomers, these building blocks, to build more of themselves, right? They go through the process of replicating, and now we've got new cells. And it doesn't matter what type of organism we're talking about. They do similar stuff, all right? But when we step it up and think about eukaryotes, it is a little more complex, just for the fact that they have partitioned and segregated specific reactions that happen in the cell to different types of organelles. The same sorts of stuff is happening in prokaryotic cells. It's just not sequestered away or partitioned in a little organelle where it's doing its own thing, all right? So kind of keep that in mind as we go through here and talk about some of the complexity with these cells. Just keep in the back of your mind, our little bacterial friends, they're doing similar stuff, all right? Just a little bit different, all right? And as we think about stuff like metabolism, although we're not going to go through great details of that, really, it's a similar idea. And the mitochondria that are producing massive amounts of energy for these cells, as we described last time with the endosymbiosis, these guys were bacteria to begin with. And so the process that they're carrying out is the same with the bacteria, right? At least with the aerobic bacteria. There are differences with that as well. And we'll hit a little of that as we bump into the next material and think about prokaryotes in more of a overview with that. So there are lots of things in here. And again, things that some we might see in a prokaryote, stuff like flagella, right? The way that the flagella functions in a eukaryote versus a prokaryote is a little bit different, right? And we showed the flagella with those prokaryotes and the fact they kind of spin around like a propeller. It's different with eukaryotes, and we'll see more details with that, whether it's at the end of this or if it's in their specific chapter. But instead of the flagella spinning around, there's a bunch of microtubules that are set up that basically move back and forth, and that gives the flagella the ability to kind of wag like a tail. It propels them this way, a whip-like motion instead of circular, all right? Same idea to move, but how they do it is a little different, all right? Lots of these things, there are individual slides that we'll go through and talk a little bit about this. I don't want to get super bogged down with all of the details, but I do hope as we look at this and as we move through the rest of this, a lot of this stuff kind of comes back. You're like, I've heard this before, right? I do feel like this really should be some review. And if you find yourself thinking maybe not so much, I recommend you hit the chapter and read through that material a little more thoroughly to get yourself up to speed, all right? So there's a lot of stuff going on here. There's a lot of arrows pointing every which way. And like I said, we'll see these blown up in their own details in a second. Just right off the bat, you can see there's a lot of stuff happening in here. There's a lot of material on the inside of these cells that, like I said, segregates things into their own types of functions. And then we start collaborating, building things. And we'll go through and see some of that. So I'm not going to spout off all of the anatomy right here. We'll see more of that as we progress along. There are all sorts of details when we think about eukaryotic cells and we think about their potentials, right? We really want to focus on things that most likely would be disease-causing organisms, but there are a plethora of other eukaryotic, single-celled organisms that aren't focus of our own disease or anything like that. With lots of stuff, when we think microbes, we've got more details with that. If it's something that causes, you know, some sort of illness, either to people or to our pets or livestock, well, we want to know about that, right? We potentially want to know about other things as well, but there's definitely more research that's poured into stuff that makes you sick, right? So eukaryotic cells, when we think about these, there's all sorts of different types of shapes. There's different types of cells that have capabilities to move or maybe not, all right? And sometimes we can classify these cells by how they move, all right? And we start thinking, categorizing like that. The flagellates, we've talked a little bit. We showed you some giardia. We've got some trypanosomes up here in this middle frame, and here's looks like maybe a flagellate right here. Not exactly sure what that is. Poking off of there, it looks like it could be a flagella. It says it's some algae. And you can see the green chloroplasts within here. So some trypanosomes, vorticella, kind of looks like a couple of bells on here. I feel like that's maybe something going on too. Paramecium, these guys are ciliates. So similar but different to the flagellates. A bunch of little short hair-like appendages covering the surface of the cell. These things are typically super small, and in this case, you can't really see it. This kind of halo around it, that's the cilia. And if we were to zoom in closer, then you'd see the fuzz surrounding them. That's all of the cilia, all right? And then the last one over here is some malaria. Malaria parasites are not motile, and Plasmodium is the culprit, the species that causes malaria. And we can see a red cell here that's infected, and that's probably some white blood cells up there. So different shapes, different ideas as far as how they are surviving, maybe photosynthesizing or other stuff, the heterotropes, right, versus autotropes. So now for some of the details of those cells, the structures. Eukaryotic cells have all sorts of complexity, and typically, if you're looking at a eukaryotic cell, whether it's some of our own white blood cells or maybe some of these single-celled organisms, if you're looking at them under a microscope, if you've stained them up, almost always the first thing that you're able to recognize is the nucleus of these cells. When we stain them, they're deep purple, they stick out. It's like a beacon saying, here's a cell, look, here's some of its material. Very easy, typically very easy to visualize. Maybe in biology labs, there's a lab where we look at, we're thinking mitosis, and we look at onion root tips. We look at some other stuff as well, but the root tips are pretty good. And as you're initially looking at these slides, you probably see lots of rectangular cells, the plant cells, with purple bits on the inside. You're seeing the nucleus. And as you keep moving down toward the tip of that root, all of a sudden, those purple things are gone, and holy cow, I'm seeing chromosomes, right? Depends on the cell, and it depends on the process of the cell cycle that it might be in as far as whether or not you're going to see a nucleus or not, all right? But typically, first thing you're really going to see, all right? Very easy to recognize. And there are, when we think about things like white blood cells, there's maybe some differences with that that allows us an opportunity to kind of differentiate and categorize as well, all right? So nucleus, and we've got the membrane-bound structure surrounding it, keeping it all intact. There is an area called a nucleolus that is kind of central in there. And this is an area that's kind of rich with activity, where we see production of RNA molecules. It's where we are synthesizing material like messenger RNA or the ribosomal or transfer RNAs that need to be produced to build proteins, all right? So there are other details with some of that as we progress through. Here we can see a couple of cells, and the green around here is the nuclear membrane. And the inside, the nuclei in the red, so some fluorescent stains, we've got DNA that's picking up that, and then the membrane, so phospholipid, picking up the green, right? And then we've got some other protein cytoskeletal stuff that we can see, kind of fuzzy, but it's there, all right? So nucleus, and then butting up against the nucleus is the part of the endomembrane system. It technically is a continuum of the nuclear membrane, and then we progress outward, right? So it also depends on the type of cell as far as what sorts of details we'll be thinking about and how much of this stuff is really going on. Are we pumping out all sorts of proteins? We're going to have lots of endoplasmic reticula. And in this case, what we're looking at here is the rough ER, or the rough endoplasmic reticulum. All of the little red dots on here are ribosomes studded on this membrane. And in turn, messenger RNA will associate with these ribosomes. And as we make those, as we start making our proteins, as we are reading the messenger RNA, the protein will stay inside in the lumen of the ER, all right? And that's important when we think about protein. There are all sorts of things that could interact with these proteins, maybe good and maybe bad. And if you're building proteins, if you're making something, you don't want to get it messed up before you've even completed it, right? You want to keep it protected, right? And so by keeping it in the endoplasmic reticula, it prevents it from getting acted upon by different enzymes that might degrade it, or different acids that could potentially tweak the way that it folds up and bends and all packages together and create something that should be functional that's now not, right? So the rough endoplasmic reticula is a mechanism to keep those proteins that likely are going to get moved elsewhere, probably shipped to the outside of the cell. It keeps them protected, all right? Eukaryotic cells also have ribosomes that are free floating in the cytoplasm. We'll see that stuff in a bit, all right? There are proteins that we are going to be making that we want to keep on the inside of the cell, all right? Maybe some of these cytoskeletal filaments, some of these other things inside, they stay inside, right? And in turn, we don't need to ship them to the rough endoplasmic reticula in order to do that. They just go right to the free-floating ribosomes, they build it, and they stay within that cytoplasm or cytosol, and they do their functions. So it truly kind of depends on the fate of that protein, right? There are other details that, you know, maybe you're asking yourself, well, how do you know? How do you know what protein you're going to be making with that little chunk of messenger RNA? How do you know it should go to the rough ER or just to some ribosomes free floating in the cytoplasm? The message has that information. And as we are producing this messenger RNA, and in eukaryotic cells, we've got to modify it. We've got extra stuff on there, and we cap it, we put a tail on it, and then we export it out of the nucleus. And dependent upon what that little bit of a signature says, it's like you're writing a letter, right? No emails here. We're going to actually write a letter to somebody. If you're shipping off a letter, you put an address on there and you put a zip code. That zip code directs you to where it truly needs to be going, right? And then the address gives you the specific details, right? So this specific little tag is like a zip code that says, oh, rough ER or cytoplasm. And as it moves out, it knows what it's going to associate with. Pretty cool, all right? So that's the idea with this. And there are, as we will see, numerous other things as we associate thinking this type of mechanism. We're not done here, all right? We need to move this material, and it's going to be packaged up in part of the membrane off of this. It's a fluid system. There's give and take, right? And so we'll keep that in mind as we progress along. We can see an actual image, an electron micrograph, nucleus and nucleolus, the nucleolus, the dark bit, all of this is nucleus, right? This is all of the nucleus. There's boatloads of chromosomal material within here. And then little bits, little pores, right, like this, as we look at the cartoon, little areas that are connected that allow for material to move in and out. Stuff needs to come into the nucleus as well, all right? So like building blocks, right? We need nucleic acids, whether it's DNA or RNA, that we're thinking about the ingredients to make more of ourself needs to be able to come in, right? So butted up against the rough endoplasmic reticula is more of the endomembrane system, but now this stuff lacks the ribosomes. The smooth endoplasmic reticula helps with other things. It functions together, all right? We see things like lipid synthesis. We also can see in some cells copious amounts of smooth endoplasmic reticula, and we think about like cells of our liver full of this stuff, and it helps to detoxify things as substances are pulled in. We can break things down and degrade them, right? So there's give and take. We can be, you know, doing things inside or producing stuff for the outside. So other things functioning within the smooth reticula And again, kind of a continuum. It's right next to the rough ER, and it's not exactly connected. Another bit is the Golgi apparatus. And Golgi or Golgi bodies are a bunch of flattened membrane systems. And, you know, I've progressed in my thinking with this the littlest bit. I used to describe it as a post office, right? Because that makes sense, right? But I'd rather describe this as Amazon, right? Now we've got a distribution center, right? We've got all sorts of different stuff, right? Almost anything you could ever imagine. And we've maybe got it all in this warehouse, right? And maybe we're on Amazon and we're ordering up some stuff. And you're like, you know what? Package it all up in one thing. I don't care if it takes 2 days extra to get this, if I have to wait for that. Put it all in one box, right? That's what you're telling them. That's what Golgi does, right? So let's take, for example, a lysosome, all right? Lysosomes are like our garbage disposals in our cells. They help to break things down and degrade them. They're full of all sorts of stuff, digestive enzymes. Those are types of proteins, all right? There's some other substances, acidic products that also help to destroy things, all right? Numerous substances. A lysosome could have upwards of 50 different types of molecules, enzymes typically, that are in there, all different proteins. And each of these different messenger RNA strands coding for this specific protein, it goes through the endoplasmic reticula, it blebs off in a vesicle from the endoplasmic reticula, the rough, and it will fuse to the Golgi apparatus. As it makes its way to the Golgi, it fuses to that membrane and it dumps its contents to the inside. Now the distribution center is receiving the packages, right? And they then take them and decide what's really going to happen. And they start packaging things up together if we're talking the lysosome, all right? Now we get all of these things and we start putting them into the same package. As we progress through the Golgi apparatus, collecting other parcels to put into my one container. And we make our way to the opposite end of the Golgi apparatus, the shipping end, all right? Receiving and shipping. And on the shipping end, we've got our vesicle that's now full of all of the contents. It blubs off in a different vesicle. Now this one full of potentially numerous products, all right? Doesn't have to be, it could just be one protein, all right? And that we're gonna export that maybe to the surface of the cell, all right? Fusing with the plasma membrane and pushing it out. The lysosome is gonna stay inside of the cell and it's gonna stay within this vesicle and it's gonna have the potential to degrade substances. It may potentially fuse with a pre-lysosome depositing all of these nutrients, not nutrients, all of these molecules that can degrade stuff, right? So all of this stuff still walled off, kept separate from the cell itself in these membrane-bound vesicles, right? So we can see more as we're looking at this and the blow up of this. Here we've got a plant cell versus the animal cell that we were looking at, but it's basically the same idea. So another image, this blowing up, showing the Golgi apparatus, a bunch of flattened little sacs within here. And we can see this up here. The cisternae are the little pieces all stuck together inside, just like the rough endoplasmic reticula. The inner bit is called the lumen. And this is where the proteins are able to get collected and packaged up together, right? Coming out in a new vesicle. So another example where there's give and take, right? We lose some of the membrane from Golgi as it is taking some of its packaged material, and we give membrane to Golgi as it is accepting materials, give and take, right? We're also getting stuff from the smooth ER as it shows up and distributes stuff as well. So there are other substances, and it does depend on the types of cells as far as some of these things, do we have them, and the quantities that we may have. Peroxisomes, as we can see in a real image, and over here, a couple of little vesicles. These ultimately allow properties for cells to deal with substances and harbor materials that also helps to potentially degrade mechanisms. But lots of these peroxisomes help to deal with oxygen, which can be toxic to a cell. Here's some more fluorescence microscopy. Again, nuclei, the purple within here. The handful of micro filaments and microtubules, actin and tubulin, different types of proteins, right? We think of things like actin and myosin for our muscles. This is one bit of that, right? And so similar sort of function when we think about that, but it's only one half of the idea of like a muscle fiber, right? So this is all really making up parts of the cytoskeleton, kind of an internal scaffolding, helping to tether things in place and also lending to the ability for some things to move along some of these filaments, like moving along this actin. Almost like a train track, allowing for substances to move from one area to the next, transporting vesicles, right? And there are some motor proteins that it almost looks as if it's a little guy walking right along there as it is carrying a vesicle, right? Kind of interesting. So here, a microfilament, we can see a pair of actin filaments together, kind of twisted around, right? And we can see this with cells like some amoebas. We could think about cells like some of our white blood cells, not all of them, but like a macrophage that might be doing surveillance within our tissues, hoping to find some sort of foreign invader and destroy it. They move by this amoeboid type of movement, pushing out their cytoplasm and kind of pulling themselves along. And it's these actin filaments that basically lends to that ability. Again, kind of like a muscle cell, right? So you can see it relaxed and contracted, lending to its ability for movement, right? We can see other things as well. Two cells in the process of dividing. The cleavage furrow that forms is the result of proteins, this contractile ring, and it's a handful of actin filaments that constrict and pinch it off, creating two distinct cells, right? There's other stuff here, the cytoplasmic streaming, that's basically, it makes me think of like a windy day, right? It's kind of the same idea, but it's on the inside of your cell. It provides this ability for the cytoplasm, the sugary water, if you will. It's not just sitting there like a stagnant pool of water. It's churning, it's moving, it's allowing these molecules to have some motion so that we can get substances moving around, and parts of the cells can deal with specific things as it's all moving rather than just one, stationary, stagnant body of water. So we can see other, as we're looking at some of these intermediate filaments, polymerized subunits, a strand almost looks like a rope as we see this sort of cord within here, all right? And these are more of a permanent situation as we can see them in some circumstances like this around the nuclear envelope. There are other potentials, these two different cells, and we can see these filaments that are part of the inside, but it helps to tether them. And this would be the space in between one cell and the next, right? And those desmosomes are really what we're looking at in both of those instances. So the microtubules, and here's the motor protein that I was saying, looks like a little guy. I know there are some videos that show some of this, and it does look like it's holding this big vesicle above its head and just walking right along this, right? As it's moving the motor protein, moving a vesicle from one location to the next. Maybe this vesicle is a lysosome that we want to move to another area, right? Whatever, right? So hollow tubes, right? And its tubulin dimers linked together like this, right? So all sorts of different potentials with that. So here we're thinking more like mitosis or meiosis, depends on the type of cell, I guess. So the ability to move the chromosomes, to partition them and separate them to opposite poles of the cell. We've got our centrosome right here and a bunch of microtubules that link up to our chromosomes, and then we will pull them apart. As we do this, we will separate and partition. Here we've shown, we see these chromosomes, and you can see some bits in here, although they all look red with little blue bits on here. We should show some blue ones in here because it looks as if crossing overs happened. I really think that's what they're trying to show, but where are the blue ones? How did we get this blue in here? That's fine. I won't judge. I guess I just did, and that's okay. All right. The mitochondria, we talked a little bit about mitochondria with the endosymbiosis. Here's some details as we look at it. A kind of bag within a bag structure, an outer and an inner membrane. This becomes important when we think about the ability to really harness some energy in the first place. There's a lot of stuff going on. And as we think metabolism, if we're thinking a eukaryotic cell, right? The first thing that happens, if we're thinking like sugar, we're going to be able to pull in other things as well, but sugar is typically what we focus on as we talk about metabolism. As sugars are brought into that cell, as they move through the plasma membrane, they get into the cytoplasm of the cell, and glycolysis, the splitting of that sugar, happens in the cytoplasm. Now we've got some pyruvate molecules, and those pyruvates are going to get manipulated a little bit by an enzyme, and it's going to shuttle them into the mitochondria. So the first bit is in the cytoplasm, and everything else happens in the mitochondria. All right. So the mitochondria, we describe them as the powerhouse of the cell. They generate ATP molecules, which are like rechargeable batteries, and the mitochondria is the charging station, right? Constantly functioning to provide ATP, right? So there are lots of things as we're looking at this. I mentioned before, they've got their own DNA. Their DNA is circular, like bacterial cells, whereas the chromosomes we see in the nucleus of a eukaryote is linear, right? They produce basically everything that they need, right? Almost everything, right? And with this symbiosis, they basically become dependent, and we dependent upon them. Our cells provide a little bit of material for the mitochondria. All right, it's basically like, why do I waste my time making this when you're already producing a boatload of it? Just give me a little of it, we'll be good to go, right? And I'll give you plenty of ATP. Just help me out a little, right? There's a little bit of stuff that our own chromosomes are producing, and the mitochondria picks up a little bit of that. But for the most part, they're self-sufficient. All right, so there's all sorts of enzymes, there's all sorts of proteins studded on this inner membrane that in turn is going to function in harnessing this energy, all right? We in turn will produce a concentration gradient of hydrogen ions, and those hydrogen ions go in this inner membrane space, right? They are pumped in here against their concentration gradient, right? And then they trickle back in through ATP synthase. And as they do that, going down their concentration gradient, that's how we make ATP, all right? So it's metabolism in a quick single slide, right? So chloroplasts, we're not going to think too much about this stuff, all right? But we do want to think a little bit. There's definitely some potentials with some algae and things like that. There are some things that are important when we think about microbes. So the photosynthesis happening in chloroplasts, this isn't the same idea as mitochondria, but it's sort of like that, right? And it's almost really kind of the opposite, right? It's not exact, but it's almost like that, right? So the mitochondria is going to be breaking down sugars and creating energy, and the chloroplasts are going to be producing sugars as they harness energy, right? So kind of backward, right? Kind of opposite. They're also a bag within a bag, right? And the these chloroplasts, this photosynthesis that's occurring inside of eukaryotic cells, there's another, as we described last time, secondary endosymbiosis. A photosynthetic bacterium was engulfed, and in turn, double membrane. Inside of it are some other structures that are really the business end of what's happening, all right? The thiocalcoid, all of these little stacks together, we call it a granum. Single little pancake looking guy is a thiocalcoid, but a bunch of them together, granum, all right? All of these things in here are full of chlorophyll pigments. That's why it looks like a green bean up here, because chlorophyll green pigment, all right? So we've got all of this chlorophyll, all of these chlorophyll molecules, and they are going to take that energy from the sun, and it excites molecules within the chlorophylls. That in turn generates some electron carriers and a little bit of ATP. But this ATP is not the energy that we're thinking about for that plant to grow. It's the energy needed to manipulate the CO2 molecules to build sugars, right? Once we build the sugars, then we flip it around to the mitochondria and we're like, all right, bust them apart, right? Now you can get energy, all right? So the light portion of photosynthesis happens in the granum, in the thiocalcoid membranes, and then they export the energy and the electron carriers into the lumen, the stroma of the chloroplast. And this is where the sugars, the synthesis portion of photosynthesis, this is where that's occurring, all right? So we showed membrane before, and we looked at this stuff and saw very similar things, right? When we were looking at prokaryotes. Plasma membrane is super similar, right? Doesn't matter if we're talking a prokaryote or a eukaryote, very similar, all right? What it's expressing, some of the proteins or sugar proteins or sugar lipids, whatever, right? Some of that stuff is going to be different, and it should be, all right? We've got our own signatures on our cells, and every one of us in this room has different signatures on our cells, right? There is some stuff that's the same, right? But other things different, right? You can't just readily give a kidney to anybody, right? Donating blood is a little bit different, and that has to do with different fingerprints that are on those cells that lends to that. We'll talk about that in the next unit of material, all right? But we can see this phospholipid bilayer creating a situation that keeps the inside in and the outside out, and potentials for stuff to move through that membrane really depends on what that stuff is, all right? Some things can readily move by diffusion through this membrane. Others need some carrier channels. And in other circumstances, through the process of active transport, we will push things into or out of the cell against concentration gradients. But similar idea as we described last time, all right? And similarly, like the endomembrane system that we were looking at with this give and take, the plasma membrane is no different. We've got the ability to accept membrane and to take some away. If we were to phagocytize, if we were to pull in some bigger food stuff, we would surround it by the membrane and pull it to the inside. And now we've got a membrane-bound vesicle, plasma membrane, that it has encased this substance. It in turn will link up with a lysosome. We start degrading it, all right? So that's this bit right here. And there are different types of engulfing stuff. Endocytosis is pulling things in. And in this process, it is in a vesicle. It just depends on what we're doing, all right? Phagocytosis, penocytosis, and receptor-mediated endocytosis, they're all pulling stuff in. It's just what stuff it is that's going to vary, all right? We think of phagocytosis as bulk feeding. We think of penocytosis as cellular drinking, right? And then this, well, that's kind of feeding as well, not really drinking. It's like this, all right? But the penocytosis isn't just drinking water, all right? Cells can deal with water through many mechanisms. Osmosis, trying to keep stuff balanced, right? Sometimes water molecules linking up on other molecules and they hitchhike a ride onto the inside. There's a handful of ways that water can be moving through. Penocytosis isn't just like getting a vesicle full of water and bringing it in, all right? Really, it's some particles. It's instead of taking a glass of water and drinking that, now we're drinking some Gatorade, right? Or something else, right? You've got a bunch of electrolytes. You've got a bunch of things that are dissolved in that water, and it's that stuff we're getting in, all right? So drinking, but more than just water, right? In all circumstances, again, it's inside of a vesicle. So here, this says large particle, but it looks like a bacterium to me. And so I imagine this is a macrophage pulling in a bacterium and destroying it, all right? And we'll go through details of what macrophages really are doing in a bit, all right? But we've pulled something in, all right? Here, we've pulled something else in, but it's considerably smaller, right? Those solutes that we're pulling in and bringing into the inside. This last one, the receptor-mediated, goes back to here, you know, where we've got all sorts of things on the surface of the cell. Some of these proteins might be receptors, things that can be stimulated to do something, all right? So this specific receptor can recognize something, all right? And maybe we could think of it, again, like a liver cell. And this liver cell could have some receptors on it that could recognize cholesterol molecules. And the liver cells, if cholesterol molecules happen to come across, it will snag them and pull them to the inside, specifically thinking the LDL, the cholesterol that you maybe want to be trying to minimize a little bit, right? And the liver cell pulls it in and tries to manipulate and deal with that. As this happens, it's like the phagocytosis over here, but we've got these receptors. So we snag our substances and potentially a couple of them, because that's what we've got going on right here. And on the inside, now we've got that along with the receptors. And so we call this a coated vesicle because it's got other stuff on the inside. It's packaged it up and held it as it's brought it in, all right? Similar idea, right? But a little bit different as far as what it's actually bringing in, right? So this more with the membrane and the extracellular. So here's the inside, here's the outside, right? And the extracellular matrix it's not the same, but it's like the cytoskeleton that's on the inside of the cell, helping to provide some internal scaffolding, tethering, and anchoring things in place or lending to some movement. The extracellular matrix does a similar type of function by connecting one cell to the next, kind of anchoring stuff in place, right? And also maybe lending to a little bit of movement with that as well. So there's proteins and sugar components that ultimately kind of act in this matrix. There are other potentials with some little bit of communication as well with the matrix. So here's an image of some flagella and or cilia, very similar, all right, when we look at them. Structurally, as we look at these microtubules, it's the same, basically same idea, and they function the same, all right? The flagella, typically, you know, a few, and there's potentials where maybe they're under the plasma membrane, creating that undulating membrane like the trypanosomes, or they're out and exposed like Giardia, all right? Or we could think of a sperm cell with a single flagella, all right? Same sort of an idea, all right? As we look at this image, we've got microtubules around the periphery, and they are doublets. So we've got 2 microtubules, and there's 9 of them, 9 pairs as we look at this. And then on the inside, we've got another pair, all right? So the 9 and 2 arrangement. And they are connected to each other, and they move like this. The two little circles together can slide like this, and they work in unison. And so you get them all going back and forth like this, and it makes the flagella or the cilia whip, right? And here's a paramecium that we showed before that looked just like a little halo kind of around it. Now we've got it through an electron micrograph, and it looks all fuzzy, all right? It's got all sorts of cilia covering it, right? Here, it's, although you probably can't really see it, this says mouth. This is a cytostome, or mouth, right? And it's full of cilia as well. And that cilia is moving, hopefully, moving foodstuffs into the mouth of our paramecium. Oh, that was good. That's exactly where we want to be, right? You're like, nope, that was too fast. Okay. Stop sharing. Bear with me. Quit this. And I just, this morning, I just posted these PowerPoints, so I do apologize for not getting that stuff up there earlier. What? All right. I will tell, before I start this, I will tell, you know, as I was doing all of these lectures, I did the recordings. I did them over the course of 2 days. And the last one I had talked, I think it was almost an hour and a half. I'm doing all of this stuff. I finish and my whole computer, it's all just frozen, right? I can't exit. I can't do anything. Can't make it work. And a little bit of fear rolls over me. I'm like, oh my gosh, am I going to have to talk again? Right? I'm going to need a bigger glass of water, right? That's not good. And I'm like, I don't know what I've done. I don't know what's going on. So what do? I just push the power button, right? Like I can't, nothing will happen. I push it. I'm doing everything, right? I can't do anything to make it work. Power it off, and then it opens back up, and it's not there, which is exactly what I thought was going to happen. Like I've lost it, right? But then I got onto Zoom, and surprisingly, it was there. I was like, how did this happen? It still gave me a couple of flags. It's like, this could be corrupted. And I'm like, I don't care. Just upload it, right? I clicked it and opened it, and it looked like it was fine. So hopefully it truly was fine. My gut tells me you probably have not watched all of those lectures in the first place, but maybe, right? We'll see. I won't ask because I don't want to see if you have or haven't. I will just keep it to myself. And no, you haven't. That's okay. You should. Okay, the prokaryotes. These next 3 chapters are like the details. It's trying to introduce you to a plethora of different things. All right, I'm going to show you a bunch of different bacteria. I'm going to show you a handful of archaea. We're going to talk about all sorts of different things. We'll bump it over to the eukaryotes and talk about protists and worms and talk about some of those details. And then the last one is the acellular stuff. Viruses are the main idea, but there's other stuff like some mad cow, some prions, and some other things as well that are in that last bit of material, all right? Things we've already kind of briefly thought about, we're going to expand upon as we move through, all right? So kind of keep that in mind. And also, as we are introducing these, a good chunk of them will be those that really are causing some disease. You can kind of expect that we're going to revisit a lot of this stuff in the tail end of the chapter when we really start thinking about what's up with these organisms, right? So our first introduction, and then we will dive deeper in the last bit of material with this as we think more about them. So all sorts of organisms, all sorts of opportunities, right? As we think about just the complexity or the diversity, the sheer amount really is pretty mind-boggling to me. When it comes to the prokaryotes, bacteria and archaea together, we're going to put them all under this umbrella. There is huge variation. There's huge diversity with all of them, right? And I like to think of them, you know, like where you think there should be bacteria, and I know you guys walked around the first lab and you did a bit of swabbing. All of those things, they take a little bit of time to grow. So you maybe have peeked at them, but you're really going to look at them a little more, I think, next Monday. You'll really look at that stuff and see it. After it's had about a week, you'll think, holy cow, right? My oldest son, he's in high school. He had to do a science project, and part of his was thinking about kind of what you guys did, swabbing some stuff, but he was thinking about just doors at the school and what sorts of organisms over the course of a week, right? So starting off, and then Friday, is there differences? Is there variation? It looks like it doesn't really matter what day of the week. That's stinking. Both, he did 2 doors. One, and it's all just the outside. Part of it is I did go with him one day just to see what was happening. And I tried to stand back, right? It's like kind of like science fair sort of stuff. I don't want to be the professor that gets in here and then the kid shows up and he's got all of this shit that they're just like, well, look at this, right? This is fantastic because I did it, right? No, I'm not doing any of it, right? I'll give you some Petri dishes. I'll get you some swabs, right? You do the rest, right? I don't want to step in because you can tell in a science fair if the parent did it or if the kid did it, right? You know, right? And so I'm not doing that, right? We did talk a little bit about some things that maybe could be good, right? But I didn't tell him anything else, right? And I'm no hand holding, right? So he has swapped two outside of two doors, one door that's pretty heavily used and another one that's maybe not, right? At least from what I gathered, right? Both doors over the course of a week's time, there's all sorts of stuff and lots of different stuff. And my gut tells me when you guys swabbed some of the environment stuff, I don't know what you swabbed, but I bet you're going to see a lot of different stuff, right? Some of it could be a potential pathogen. Some of it could maybe make you sick. He's got boatloads of different types of molds that are on there. You're probably going to find mold, right? Mold spores are all over the place. You could take a Petri dish and just open it up and leave it exposed. I put it clear over there. So I'm not sitting here talking, right? Not wearing a mask, right? You are 2 rows back because you know, you're like, I bet this guy's, he's going to spew some spit out, right? That's not really why. So you're going to find all sorts of things. And even if it was just the open air, mold spores will fall down, bacteria floating around on dust particles, they may land on the plate. That stuff would grow, right? And if you left the lid off for a half an hour or so, you'd probably get some good stuff. If you just open the lid up for 5 seconds and closed it, well, maybe you're not going to hardly find anything, right? Give it a little bit of time, you'll probably find some stuff on there, right? So I've seen all sorts of things on these plates, and some of them I'm just like, holy cow, that's awesome, right? Or gross, right? Depends on how you feel about it, right? And that's as far as we've gone. We've, you know, and I'm not spouting stuff off like, oh, it's this or it's that or what. That's not my job, right? And that's not really what he was setting out to determine in the first place. He was just looking at a time course, right? And what do we see, right? So it's a long story, right? When I think about organisms like prokaryotes or just microbes, right? But we're in prokaryotes right now. Where you think there should be bacteria, well, there is, right? Where you think there's no chance there could be bacteria or prokaryotes, I think you might be sorely mistaken, all right? Places that you would think super inhospitable or very dry or whatever it could be, too salty, right? It could be there, right? And maybe not living very well, but potentially living, all right? You guys maybe know some Himalayan sea salt, the pink sea salt, right? Looks kind of cool, tastes like salt, right? There's no difference between it and regular salt, or is there, right? The pink Himalayan sea salt, it's pink because of a type of microbe that's within there. And, you know, it's really an archaean, but halobacteria is its name because it was named that before we ever separated it out into the archaea, which really makes me think, you know, we change all sorts of names, but this one, I'm like, what's up, right? Can't we just call it haloarchaea because that's who it is? These things, like salty environments, you can see some of this stuff in the Great Salt Lake. And in the course of the summer, as we start progressing, we'll see some halo blooms. The lake kind of looks purplish pink in some regions. And that is the result of these microbes, right? The Himalayan sea salt is the result of the microbes within there. And you could technically get those to grow if you wanted, even though they're entombed in the salt crystal. They can survive molar concentrations of sodium chloride, like 5 molar. That's a crystal of salt. And I saw an article a day or two ago. They speculate, I think it was like 800, it's maybe like 800,000 years old or something like that. They think these microbes encased in this potentially are still viable. Right? That's impressive, right? Whether or not that's true, I don't know, right? So that seems pretty unlikely, right? Could you live in this salt crystal? Could you live in this inside of this rock? Are you living, right? Interesting, right? You could, you know, take a core of the earth and go down almost a mile, and it's likely you're going to find microbes, and it's not contamination as we've drilled down there. It's microbes that are down there, right? Super hot, super cold, acidic, basic, any sort of thing that you're just like, you flat out can't live there. Probably can't. All right. Shewanella lives in the deep sea, all right, near hydrothermal vents, right? It's got these nano cables, kind of like thinking that, you know, the ability to conjugate through this pilus. It's a similar idea, but slightly different. And these are like, it's almost like you're out there fishing, casting out your line. The nano cable is basically fishing for some oxygen molecules, snagging onto that so that we can in turn metabolize a little bit, all right? And that's both images with it, but we can see a handful of them, all right? And this, you know, is the Dead Sea. I feel like you know, if I was on my game, should have a picture of the Salt Lake here instead of the Dead Sea because it's a little more relatable to us. But we can see a little bit here with some of the color around there. I technically flew over the Salt Lake over the weekend. And you can see a couple of areas where they've walled it off where Morton Salt Factory is getting their salt, right? You can see a few of those areas and there's different colors within there. And it's really some of the archaea that's imparting some of that as you look at it. Lots of it's white, right? So that's what we're seeing here. Halobacterium salinarum likes the saline, likes the salt, right? And halo is a salt lover. Halophiles are salt lovers, right? So this giving us some of that. And, you know, another thing with the Salt Lake, way back, I don't know how far back, 1940s maybe, the Army Corps of Engineers built a railroad track that basically cuts the lake in half. When they did that, this train track that goes through the lake doesn't have any sort of coverts underneath. It doesn't allow for the water from the north part of the lake to mix with the south part of the lake. Salt Lake is technically two lakes, right? Cut off by the train track, right? And as this train track goes through the lake, if I remember properly, the northern portion of the lake doesn't get as much fresh water coming into it. The southern portion gets some more. So the northern portion with not as much water coming into it gets more evaporation, more concentration of the salt in the summer months than the southern portion. That gives you a bigger bloom. And the northern bit looks more pinkish purple than the southern bit, all right? And if you watch the weather in the summer and they're showing satellite images, you can see the Salt Lake and it's two-toned. And it's two-toned because of the railroad track that ran through there, that cut it off. And we've got different blooms of archaea, I want to say bacteria, different blooms of these guys, right? Crazy and pretty cool, all right? So you'll watch the weather and you'll see it and you go like, oh, right? The astronauts on the space station can see it, all right? That's pretty stinking cool, all right? So now I'm like, dang it, why didn't I put that picture up there, right? Now you're gonna have to watch the weather, right? All right, here's some other bacteria that are, I think, pretty cool. Nitrogen fixers, like rhizobium, we form these little root nodules in some types of plants, all right? Some legumes, clovers, you can see peas and beans that are nitrogen fixers, all right? Where they've got these nodules and that in turn allows them the ability to basically harness their own fertilizer, right? Nitrogen is something that is self-limiting with plants. You need to have nitrogen for growth. And if you don't have enough, plants aren't very happy, right? But if you're making your own, if you're getting your own nitrogen, right? it's a good situation, all right? So here we can see these bacteroids, bacterial-like cells that are basically fixing some nitrogen, and these are within the plant itself, right? Another symbiotic relationship. The plant's providing nutrients to the microbe is providing, well, nitrogen, nutrients to the plant, right? And nitrogen, in the atmosphere as nitrogen gas is useless to us. We inhale it and we exhale it, all right? We can't harness that nitrogen. We get our nitrogen through things that we eat, right? And in turn, microbes manipulate it from the atmosphere. They fix it, whether it be in plants or just microbes in the soil, right? They fix it into nitrogen that's usable by plants. And then in turn, denitrifiers are different microbes that will take nitrogen coming out of our back ends or our dead bodies, and they metabolize and break it apart and release it as nitrogen gas. It's part of the nitrogen cycle, and it relies on microbes to fix and to release, all right? Pretty impressive. So here we could see, again, just more diversity with these guys. Rickettsias, these types of bacteria, little rickettsial bacteria, and we're looking at hemolymph, which is basically the blood of a tick, and here's some blood cells. This is, I think, pretty interesting. We think of our own red blood cells. They're not nucleated, but in ticks and probably other arthropods, they are nucleated. So you can see a nucleus in this red blood cell, right? And you can also see all of the little rickettsia, right? So this one causes Rocky Mountain spotted fever. That's this type of rickettsia. And ticks have opportunities to transmit all sorts of things, right? We'll go through some details with those guys a little bit later. Here is another bacterium. Chlamydia is, well, there's numerous types of chlamydia bacteria. This one is an obligate intracellular parasite. It has to be in a cell in order for it to grow. You can't put it on a petri dish and expect that you're going to get anything out of it, all right? It's got to be in a cell culture, all right? So it's different, all right? And there's all sorts of potentials for some microbes that need different things one way or another to help them grow. Some of them need to be in a cell, all right? So this is just showing a cycle of chlamydia. And again, we've got eukaryotic cells that they're infecting. We've got, if we could start right here, elementary body infecting this cell. That's what we're seeing right here, a normal naive cell, uninfected. The elementary bodies get into the cell. Once they're in there, after about a half a day, they form these reticulate bodies. They're inside of a vesicle because we've pulled them in through phagocytosis. And now we've got these reticulate bodies on the inside. They start to grow and replicate, forming this big inclusion. This is about a day after they have infected this cell. 2 days later, we've got within these inclusions a bunch of elementary bodies. So we've replicated and basically matured, if you will, forming a huge vesicle inside of the cell that basically is going to cause it to die, to lyse, to rupture, releasing these elementary bodies to go on and infect more cells, all right? And the cycle continues, all right? Basically, as it's proliferating and growing inside, it robs the cell of its nutrients. It basically steals all of its energy. And as soon as the ATP is basically depleted, it opens up to go on and infect a new cell to get more energy out of it, all right? This image is Neisseria meningitidis. We'll describe this guy later on as we think about like some of the nervous system infections. Meningitis is bad news, all right? And if you present to the clinic with symptoms of meningitis, this is immediately what they go to, all right? You hope this isn't what it is, but at the clinic, this is what they think it is, all right? And they treat you immediately, trying to prevent this from killing you, all right? And if you don't receive treatment very rapidly, and it is this, you're not going to survive, all right? There are some mechanisms we can, through vaccination, we do have some mechanisms, but it truly kind of depends on those circumstances. Meningitis is bad news, all right? There are other bacteria that can cause meningitis. There are viruses that cause meningitis. And deep down, you're hoping it's viral, right? Viral meningitis, it's bad, but it's nothing like this, all right? Definitely survivable, all right, in those circumstances, whereas this could be your death sentence, all right? Neisseria is a type of bacteria that we call gram-negative, and it's a diplococci. Two little cells typically would be seen together. There is some variation with some of the Neisseria as far as how these arrangements go. There are others as well. Neisseria gonorrhoeae causes a sexually transmitted infection. That's probably more familiar than meningitis, all right? You don't want either, all right? So these, the Neisseria species, are sensitive, all right? They can't just grow on anything, all right? They require specific nutrients. This is a type of media called chocolate agar, but it's not chocolate, all right? Chocolate agar is blood agar, usually sheep, but there's other animals that we may use. Cow is pretty good too. The agar, the blood in this case, is cooked. And when you cook that, as you heat it up, the red cells get destroyed. They dump all of their guts to the outside, if you will, and it's the nutrients inside of there that these guys want. But they can't get it unless you explode them for it, all right? And in turn, they grow very well on this type of media. If you had a regular blood agar plate, it hasn't been cooked up like that, they're not going to grow. Even though they're on blood, they can't get to the insides, all right? So it doesn't grow like that. As I see all sorts of crazy media, all sorts of different ways to grow things, my first thought is, who figured that out, right? That is weird, right? Little white raised up colonies on this plate. This is our Neisseria. And again, specific factors. There's other bacteria as well that really want a chocolate agar plate, all right? So we'll think more about that. Again, definitely more about it as we get toward the end of this in a few weeks. All right, here's a radiograph. We're looking at some pneumonia, and we see some of this infiltrate within there and the caving formation within there with, it's not saying it here, but I believe we're looking at tuberculosis and not Streptococcus pneumoniae, if I remember correctly. This is pretty awesome, right? We had so pre-pandemic, we had this program called Science Trek, where I think it's third graders will show up for like a night at the museum, and they do science stuff, and they would spend the night, right? But they would interact with all sorts of different things. They're like, oh, I want to do this or I want to do that. They'd come up for a couple of hours, do some sort of science stuff, and then they'd go back, right? So I've done this a couple of years, pre and post pandemic, right before, and I was like, oh, this is super cool. Once I learned I don't have to be there all night long, right? My job is only like 2 hours of dealing with these. I'm not the babysitter, right? Once I learned that, I'm like, sweet, I'll volunteer for that, but I'm not staying overnight watching a bunch of third graders, right? That bring bags of candy and we're not sleeping, right? So this last one we had right before the end of the last semester, we had a bunch of different bacteria, some of it glowing in the dark. And we had some of this stuff. Unfortunately, these are also really pretty sensitive and they don't just glow always. And so it also needs to be almost like a complete darkness, all right? Like pitch black in order for this stuff to really function. And if you've got a little bit of light, it's not really going to work. At least that's what I learned afterward, because this didn't work very well, all right? So Alabibrio fisheri, it's a bioluminescent bacterium. It glows, all right? It lives in a mutualistic relationship with some organisms like squid. I think some octopus also has these guys within it. And this, I know there are some images with squid and this bioluminescence with this relationship. And I believe there's some sort of stimuli in between. The squid, it makes me almost think kind of chameleon-like, where it can change colors, it can do different things, but chameleons aren't glowing in the dark, right? The squid can, with the bioluminescence, it's like, you know, this glowing billboard, like, hey, here I am. Hey, ladies, come visit. Or some predator and it turns on a different color and it's like, get away, right? Something like this. It has the ability to utilize this and to turn on and turn off this luminescence kind of at its will, all right? Which makes me wonder, why didn't my Petri dish work for me? I wanted it to, right? Kind of crazy, all right? And you can see these kind of pulsating with color, again, kind of stimulating something, right? Maybe a partner or maybe a warning, right, to get away. Pretty impressive, right? And there are others where we could see some of this as well. There are some bacteria that produce some pigments that, and this is just fluorescing by itself, right? There are others where you expose it to a black light and then you can see it glowing. So fluorescence, but not bioluminescence, where it's just glowing on its own, right? And we had some of those, and those were definitely easy to see. Where, you know, it's all kind of greenish to begin with, and then it's, you know, black lights on there and it's glowing, right? So this organism, Legionella pneumophilia, it causes Legionnaire's disease, and I think we mentioned it in one of the previous lectures. This is something that is, we would classify it as a new or emerging pathogen, right? There are others that we may classify as re-emerging, like tuberculosis, something that we've known about since pretty much for forever. And then with antibiotics, we thought we had a good handle on, and now we know we don't, right? These are things that really pretty recently discovered as a potential issue for us. It was a few decades back, mid-70s, when it was really first recognized. It is associated with moist environments, water, you know, like air conditioning units, water, like the misters at supermarkets that spray on the produce. They've recognized Legionella from there. That's a potential terrifying thing. And other opportunities as well. There's also potentials for these to colonize and live within some amoeba that would be thriving in this aquatic environment, and then potentially causing issues within us. This, as I see this image, it makes the first thought that pops into my mind is that's hamburger, right? It's what it looks like, but it's not, right? Well, it sure looks like it. This is an electron micrograph. We're blowing it up, and I assume we've also got some artificial coloring in here to make it actually show up. This is Salmonella typhi. It causes typhoid fever, causes some intestinal distress, all right? You guys, I assume, have probably heard of Typhoid Mary, right? So that's this, right? And there are definitely some potentials. She was asymptomatic. She had no clue that she was making people sick, all right? She didn't feel sick and she didn't think she was making people sick. And then when they got her and told her, this is what's up, you know, you're making people sick, you ought to wash your stinking hands, right? She was basically like, screw you guys, I'm not doing that, right? You can't tell me to wash my hands. I've been making burgers for years, right? It's all fine and good, right? And in turn, numerous families had her as the, you know, cook and got severely ill. And she caused all sorts of issues before, you know, and basically told, you can't do this anymore. And she just moves and continues. She's like, I'll go over here and I'll cook for these people, right? You can't stop me. I gotta cook, right? Kind of crazy, all right? There's all sorts of details about her. So this salmonella, there's again, and there's also other types of salmonella, and some of them we think of like serovariants. So you produce an immune response, and we can detect some of it based off of some antibodies. Oftentimes we think of these as potential foodborne illness culprits. And I usually, although I said, oh, this looks like hamburger, when I think about salmonella, I typically think about poultry products, eggs or chicken, something like that, that sets the stage for you possibly to get sick. We definitely have seen a higher uptick with some instance of salmonella where people have decided they want to you know, get their own eggs. They get some chickens and they have some backyard chickens. They're getting their own eggs. They think it's great. And I think it's great too. Fresh eggs taste delicious, all right? But there are issues, all right? And there are things that eggs, you know, we deal with eggs. If we're thinking commercial stuff, you go to the supermarket and get eggs. These eggs have been washed. Your backyard eggs, you technically don't have to wash them. If you wash your eggs, you have to refrigerate them. And if you don't have to. That creeps me out a little bit, seeing a bowl of eggs on somebody's counter. And they're like, oh, do you want an omelet? And I'm like, not from you, Typhoid Mary, right? But they're technically okay. There's a film, right? That chicken lays its egg out in the barnyard and it doesn't spoil, it doesn't go bad, right? It's gonna grow up and hatch and there's a little chick, right? If you don't have a rooster, you've just got an egg, right? It's no different and it would be just fine, right? Stays good because it's got this coating and it prevents it from spoiling, I suppose. Once you rinse it off, you've basically taken that away. And now if your eggs are just left out, they're gonna spoil, right? Kind of crazy, all right? So with that, there's potential contamination on those eggs. And whether even the supermarket, if they've been washed or not, you could still get salmonella, all right? There are other issues with that. And the egg, as it is forming in the chicken, before the shell has formed around the egg itself, the inside of the egg, before that shell has even formed, salmonella can colonize the inside of the egg. So it's not just the shell, it's not just careful cracking that allows you to consume some raw eggs because I didn't get any shell in there, right? It could have been in there and not on the outside, all right? So think about that. Don't let Rocky mislead you, right? Eating a bunch of raw eggs is maybe not the best thing to do, all right? My son told me that he runs track. He's like, there's this kid, every morning for breakfast, he eats 6 raw eggs. And I'm like, what? That's kind of gross, right? But, you know, whatever. And he's like, he cracks them all into a cup and just drinks it. And he says it's gross. And I'm like, well, then why the hell does he do it, right? Because he needs the protein. I'm like, you could get the protein if you cooked them, right? Right? You could, right? Maybe not easier to just drink them down, right? But gross, right? I'm like, do you want me to fry you up an egg? I'll do that, but I'm not going to stand here and watch you eat it, raw egg, right? he's not doing that because that's gross. All right. I think it's gross, right? It looks like, and we see fruiting bodies, we see sporangium. This looks like mushrooms, right? But it's not. This is myxobacteria, and they form structures that are very reminiscent of some mushrooms, although they're small, right? Similar sort of an idea, all right? Same but different, all right? And these myxospores will in turn be new bacteria that can grow, right? And I don't know what they're growing on. I will say this is soil, although it looks like maybe some feces. We'll just flip away from that. All right, Helicobacter pylori is pretty impressive. This bacterium can live in your stomach. It causes gastritis, can lead to ulcers, and potentially stomach cancer if it's not dealt with, all right? Ulcers are an issue, all right? And ultimately, Helicobacter is the culprit, all right? There are all sorts of treatments and things like that for issues, gastritis and ulcers and whatever, but really the root of this is these organisms, right? And treating with antibiotics and some antacids, maybe a jug of Pepto, over the course of about 2 weeks, you can eliminate this organism and you can cure yourself of these issues of, gastritis or ulcers, right? They're fun little fact, fun little story. A couple of doctors ultimately recognized Helicobacter was this culprit by taking it upon themselves. They had some gastric secretions from a patient that had ulcers, right? They collected some of this fluid that harbors the bacteria within it. They grew up a big flask full of the bacteria and poured it into a couple of cocktail glasses, cheersed it up and drank them down, right? And both of these doctors contracted gastritis. They got, I don't know if they got ulcers or not, but basically they were kind of doing Koch's postulates, right? They isolated this stuff from a patient, they grew it up, they ingested it, they got the same signs and symptoms, done deal. They showed this bacterium causes this stuff. You should not do science on yourself, right? You really shouldn't, all right? And as you're thinking about going, most of you probably some sort of medical stuff, you also shouldn't self-diagnose, all right? WebMD has all sorts of things out there, and, it doesn't hurt to check some of that stuff out. But you shouldn't immediately just look at that, because almost always when you look something up, you end thinking, holy cow, I've got cancer, right? When you probably don't, right? So don't self-diagnose, right? You can get some hints and then in turn, go to somebody that actually can tell you, right? Kind of crazy. There's all sorts of details with these as far as how they can live in your stomach, neutralizing the acids and things like that lends to them getting a hold on you. But the stuff I think is cool is, those two clinicians that inoculated themselves with it. Kind of impressive. All right, here we've got some spirochetes, our little corkscrew looking bacteria. Twisted up as we see here with these axial filaments that allow them, kind of like a flagella, to whip around through their environment. It allows them to corkscrew or spin through their environment. And there are handfuls of types of spirochetes. We're not going to get bogged down with that. These bacteria, rod-shaped or bacilli-shaped bacteria, Bacteroides, these are about 30% of the normal flora in our gut. They make up a lot of them, all right? If we're talking healthy situation, you can definitely deviate and tweak that where the percentages may drop dramatically, and then you've definitely got some potential issues, all right? But Bacteroides is one of the main players, and it helps in a normal situation with metabolism. When we think about a little fresh hatchling, some lactobacillus and Bacteroides together are super important. As milk is being consumed, it helps to get it to kind of clot it and curdle it up a little bit so that it doesn't just quickly like run right through them. It gives that little fresh hatchling, you know, a little more time to digest and get those nutrients out of it in a normal, healthy situation, all right? And with that, a lot of this, as we think with that infant or with that, you know, baby, right as it's being born, it gets colonized. It picks up organisms like Bacteroides or Lactobacillus in transit through the birth canal. It picks up organisms as it makes its way out into the world. It also starts to become colonized, but a lot of the initial colonization is in transit out of the mother, and it picks up organisms. And there are definitely differences that we could see between children that were born vaginally versus cesarean section, where, you know, C-section, cutting them out of the mother, or vaginally, where they've got the opportunity to pick up microbes in transit out. Different stuff, different colonizations, different things going on, all right? That's interesting as well, all right? So we'll think a little more about that and microbiomes and all of those details later on, all right? Planctomycetes, a couple of images of these, two different, same but different, all right? Here we've got a holdfast, kind of a root-like structure that allows them to tether or anchor into their environment. And then the swarmers, these have the ability to move, but they don't have the holdfast. And then it makes me wonder, is the holdfast down there and they're not just not showing us? They don't have a holdfast, all right? So it could be, I think, a situation where maybe we're tethered down to the bottom of this, you know, pond, or maybe whatever, well, you know, aquatic environment. We're right here. We've got ample nutrients, and then maybe those nutrients start to be a little more depleted. It transitions and the holdfast degrades. Now we've got this flagella and we swim away. We try to move to some other area, a new portion of that little pond that maybe has better opportunities, right? The grass is always greener, right? So the ability to move and potentially find a better location. This, what we're looking at here is Winogradsky column. This is a tube, can do it in all sorts of things. You can even use like a 2-liter soda bottle and produce a Winogradsky column. The idea with this is to kind of, doesn't have to just be environmental stuff, but it oftentimes is something like that, where we think about maybe microbes that are present either in some water or in the soil, and we want to give them an opportunity to kind of amplify without putting them on a petri dish that you hope they're going to grow on. You know, some stuff, if you were to go to the Portneuf and get a water sample and come back to the lab and put it on a petri dish like nutrient agar, you're probably going to get some stuff to grow, all right? But that nutrient agar is full of all sorts of good stuff, all right? Lots and lots of nutrients, all right? And there are probably, not probably, there are organisms in the river or wherever, they don't have that ability, right? They're not constantly bathed in sugars, right? We're not getting all of this stuff, and providing them that opportunity is going to overwhelm them and they'll die. They can't even live in that environment. You need to grow them in minimal media. It's got hardly no nutrients, just a little bit of minerals, and they grow, but typically really pretty slowly because they're not pulling stuff in the same way. So Winograd column is kind of like that. We're not giving them lots of stuff. We're giving them what was already there. Some little bit of soil, a little bit of water, and then you potentially impart some other stuff, right? Maybe some carbon source. Maybe we want to see different types of carbon sources or different types of things that we may put in there. You can put stuff like a nail, bit of rust, something like that to see, do these bacteria want iron? Can they pick it up? Can they pull it in? It's not something that you get to show up like we're seeing right here overnight. This potentially takes months before you actually start seeing this stuff, all right? Maybe a little quicker, all right? And within this column, there's all sorts of stuff that starts to show up. Different types of bacteria, green and purple, although you can't see it very well. The purple bacteria provide this beautiful purple color within the column. Different microbes all associating maybe together and maybe not, right? Kind of depends on the circumstances. And there's other players within there as well. So it gives you an opportunity to really kind of get an overview of what sorts of organisms might be in this specific habitat, all right? There are a handful of Winograd columns down in one of the micro labs, not the lab that you guys are in, but I will try to bring those over to the lab that you guys are in so you can see them. They're in some big cylinders, and each of them, this was a, I think, microbiology class. I can't remember exactly which one it was, where they put some different things in there just to see, will they function, will they not? What sorts of things will I get? There, are examples where they put, a dead squirrel in there, or an egg, put a whole intact egg, hard boiled or not, right? And what happens? And again, I'm saying months of time, you're going to see all sorts of different things. There are other opportunities where you can go in and sample stuff. And I know, I like seeing it like this, right? But you could take a big glass rod and you could get in there and just stir that up, right? You're just playing in the mud, right? You could mix it all up, and now you're taking some of the purples and you're mixing them with the greens. What sorts of associations do you get when you interact like this? All sorts of different things. And you give it a little more time and make some more samples, right? Kind of crazy. I recall a professor's lab, a student, I believe, built this Winograd column, soil and water and whatever, and he'd had it for quite some time. And on the surface of the Winograd, you've got all of the soil, and then on the top is water. So you're creating kind of an anaerobic environment underneath. It's like the bottom of the pond, right? And you're looking at this core of soil. On the top of the soil were some algae, not the green bacteria, but actual algae, and it looked like a just green lawn, right? This nice little green lawn right on the surface. And then there's purples and greens down below and some other stuff. And on the surface of this little algae on the top of the lawn are all of these little things swimming around. Water fleas, right? Those things, whether they were in the water, maybe from the port enough, maybe they were in the soil as little eggs. They hatched over the course of time, and then there they are up on the top, and they're feeding on stuff like algae or bacteria. All of that stuff is there, right? It's everywhere, right? That's what we're getting at, right? The diversity, right? Pretty impressive. Here's algae, maybe not so good, right? We've got Microcystis aeruginosa, a type of cyanobacteria, first one of the early photosynthesizers, and you see them in freshwater environments. These guys can cause some problems, right? And the cyanobacteria were called blue-green algae. So they're cyanobacteria, right? They're not algae. They can cause some problems for sure. And here we can see this waterway that's really full of these organisms, a bloom, all right? If we're thinking ecology stuff, there are situations with like agriculture. It could be your own backyard. If you over-fertilize your lawn and water the crap out of it and then it runs down the gutter, all of that stuff's going to get into a waterway like that, all right? Now we've provided copious amounts of fertilizer, nutrients for stuff, right? And the cyanobacteria are like, sweet, I want that. They grow real rapid, they replicate, they proliferate, and they take over, all right? And you might think with the potential for photosynthesis, well, these guys, maybe that's good, right? Pumping out lots of oxygen into this water, but it's not what happens. And as they're growing real fast, they basically deplete it of nutrients, deplete it of oxygen, and any fish that wanted to say, I want to live in this water, suffocates because the cyanobacteria robbed it of all of the oxygen, right? So we call that eutrophication, where you get excessive runoff of nutrients, and you get algal or cyanobacterial blooms, because algae can do it as well. Typically, we got both of these guys in there in these circumstances, and it creates a situation that becomes very inhospitable to anything else, right? Kills it all, right? That's a problem. There are handfuls of areas where, you know, farmers, their runoff might be draining right into a river. There's a number of them around American Falls that have set up wetlands where the water that would be going right into the river or the reservoir goes into a wetland first. And there's all sorts of vegetation that helps to pull in these nutrients and toxins to prevent this stuff from happening. And you get all sorts of plants flourishing that in turn sets the stage for other types of wildlife, waterfowl and things like that, that show up different, right? And then the water coming through that wetland has basically been purified, if you will. So you don't see this eutrophication happening. Kind of depends on the circumstances and the ability for that to happen in the first place. Okay, here's a handful of other images. Actinomyces israelii, this is another electron micrograph and some false staining. They create a palisade forming, oh, that's the, sorry, Corynebacterium, it looks palisade-like. I want to say these guys we can see maybe in our mouths, actinomyces. There's a handful of other places where we may find them. The palisades is, this is with our diphtheria, Corynebacterium diphtheriae, causing diphtheria. And we call it a snapping division, so it looks kind of V-like as it makes these palisades. Not the best. Usually when we try to show this, you just see a whole bunch of them like that. We got one. That's okay. Maybe two. And then Gardnerella. This is a gram variable. When we say that, it basically, it's like, whatever. Today I'm purple, tomorrow I'll be pink, right? You get variation with them. And it's issues with their cell walls where they alter with that, right? With the way that they're picking up the gram stain. So some are cut and dry, and you can see purples or pinks, positive or negative respectively. And with others, you get some of this. There are some others that give you a bipolar staining. The tips of the rods are purple, and the centers are either pinkish or not stained at all, right? That's not Gardnerella that does that. So Gardnerella vaginalis causes vaginosis in women. Here we've got an image. So we've got some epithelial cells from a pap smear, and then some of the bacteria within there. And I believe there are, we'll see later, an image with a few of these cells, and they look very different than the other cells, and they call them clue cells. As a result of the bacteria changing the way that these cells look. I think we can see a few here and there. Not the best with that. So we talked about the Bacteroides as the big player in your gut, right? And there's a handful of other bacteria, and there's other stuff as well that are, you know, some archaea, there's some protists that might be living in there. There's a plethora of virus that you would find in your intestine as well, all right? Not virus that's causing illness in us, but bacteriophages, virus that's targeting different types of bacteria, all right? There's lots of stuff. When we think about our microbiome, what do we really have going on? Clostridium difficile is a type of bacteria that lives in your gut, but in pretty low levels, all right? If you've got a good GI tract, you got lots of good guys going on in there, the Clostridium is very low, all right? Because the other ones keep it in check. But if the situation presents where the good guys get wiped out, you're taking antibiotics for something, right? Probably not GI issues, something else. But then in turn, it creates GI issues because of that, right? So usually Clostridium difficile results through antibiotics, right? And as we're taking these antibiotics, your normal flora, your good guys get depleted. The Clostridium has lots of resistance, and the antibiotics that you're taking for whatever else don't wind up targeting these guys, and they proliferate. That lends to some pretty serious GI issues. And with a name like difficile, these suckers are very difficult to get rid of, right? Very, very nasty, all right? And so there are, you know, colitis, diarrhea, all sorts of issues with it. You're pretty sure you might be crapping yourself to death, and maybe you could, all right? Not so good, all right? There are definitely opportunities where this may warrant a fecal transplant, where you find someone that is healthy, right? And they give you some feces and you take that and it becomes your own, right? Giving you a leg up, right? I'm thinking poo sandwiches, but it's not, right? But it could be, right? This is another fun fact. As we're learning more and more about the microbiome, as we're learning about how organisms interact, we think about all sorts of things like how we were colonized, the types of bacteria that we've got going on, and that causes different types of behavior. There's all sorts of different things. They say you are what you eat, right? And it's really kind of true, right? We are feeding our friends within our guts, and what we are feeding them helps to kind of dictate who our friends really are in the first place, right? And maybe by eating specific things, it may help or hurt some of these friends, right? So prebiotics are things that you might take that help your friends you've already got going on by giving them the good stuff that they need, right? And it gets their populations to thrive, right? Probiotics are bacteria that you're taking that help to kind of give you some extra stuff, all right? So they have recognized all sorts of things with what you've got going on. And I mentioned before, vaginal versus C-section births, differences in that. There's other stuff, right? And with some of these fecal transplants, it's really pretty interesting. There have been people that needed a fecal transplant, and the person that needs the transplant was slim, right? They weren't obese or overweight, but the person that donated the feces, and usually it's a loved one, it's usually a close family member that you're getting this stuff from. So the person that donates is overweight, all right? You are what you eat, right? So now this person's profile of microbes is different than the other person's, and that person now gets this fecal transplant and was slim and trim, and they put on the pounds, all right? They become overweight, and they're overweight because of the profile of bacteria that they just got transplanted with. And the exact opposite has also been observed. You've got a person in there and they need a fecal transplant, and this person was overweight or obese. This obese person gets transplanted from feces from a person that was slim, and the overweight person loses the weight, all right? I don't want to say we have just found the weight loss solution, right? And everyone starts thinking, what slim friend do I have? And could I really befriend them and get a little poo sandwich out of them, right? Do you think could I have a little of your feces, right? A true friend would give you some stool, right? That's how I feel, right? A true friend's gonna give you some poo, right? Just to drop those pounds. I don't know. People have done crazier things, right? Back in the day, you technically still probably could, but back in the day, a weight loss fad was to order up some tapeworm pills, and you get these tapeworms, you ingest the tapeworms, you drop some pounds off, and then you take some anti-helminthic drugs, you kill the worms off, good to go, right? That is a terrible way to do it, right? And in turn, there's a lot of stuff happening with parasitic infection. And if you've got a buttload of worms, right? Those worms are robbing you of nutrients, and your body says, I'm in starvation mode, right? I'm starving to death because even though I'm eating, I'm not getting those nutrients. I'm feeding my worms. So when you're in a starvation mode, when you consume some food, your body is like, holy cow, I'm dying here. I'm going to store this as fat. I need to store this stuff as fat, so I've got reserves. So now you've got this overweight person that took some worms to lose some weight. They drop some weight because the worms are eating their food, but their body says store fat. So then they crap the worms out, curing themselves of that. They have dropped some pounds off, but you're still in starvation mode. And you keep eating those cheeseburgers and you store them all up as fat, and the pounds come right back, right? And so it turned into a vicious parasitic cycle of weight loss, worms, back to you just have to keep ordering it, right? Ill-advised, right? Although you probably could still find some, right? You can infect with worms for other things, and we'll touch on that later, all right? I do think it's interesting, all right? But the fecal transplants are pretty impressive, right? And we are learning all sorts of stuff about it, more than just the slim or overweight based off of that stuff. There's other behavioral things as well, right? And you're like, what? Why? Right? It's true, right? You know, you've got all sorts of nervous tissue within your gut. They call it, it's like your second brain, right? And these things, again, you are what you eat, right? It stimulates all sorts of other things besides your desire to have a donut for breakfast versus a bowl of oatmeal or whatever else it might be, right? There's other things. And maybe some mental illness could really be the result of what you've got populated within your intestines. And tweaking some of that stuff can open up avenues for alleviating some of those issues, right? Crazy stuff. And I really feel like we're kind of the tip of the iceberg in understanding this. This is a blood agar plate, Streptococcus pyogenes. You've all heard of strep throat, and probably most all of us have had strep throat at some point in our life. It's a gram-positive coccus in long chains, the strep, and these nice long chains of streptococci, purple coccus bacteria. On the blood agar plate, they've got specific enzymes that allow them to feed on those red cells, to destroy them by themselves. Neisseria can't do it, but streptococcus can. And it breaks the red cells apart and it sucks up all of those nutrients. We call them beta hemolytic because they can completely lyse the red blood cell, getting those nutrients, sucking them up. So you get these little halos or clear zones around them where the red plate in the blood agar is opaque. You can't see through it. And then once it's hemolyzed, it looks kind of like the nutrient agar plate that you guys have all seen already, where you can kind of see through it. It's yellow, kind of clearish, right? Different, all right? And you know that you've got something that we would describe as a potential pathogen, right? If it's beta hemolytic, if it's got the opportunity to feed on blood, it's got the opportunity to cause some issues in you, right? And if it's growing in your throat, it causes some issues, some pain, right? You know, right? You know when you've got a sore throat, right? And you may open your mouth up and look in the mirror and see some stuff at the back of your throat, maybe some whitish growth, maybe some redness, but a little bit of white patches, something like that. And you're like, wait a minute, that looks like maybe some bacteria back there, or pus, right? Pyogen is a pus producer, right? And so you can see some of that stuff at the back of your throat. And then you're like, maybe I ought to go to the doctor, right? These guys don't like salt, and we'll look at some different types of plates. Staphylococci may like it, streptococci don't. Gargling with some salt water if you've got a sore throat may help with this, because the streptococcus can't really tolerate that stuff. So you can create a situation that becomes a little inhibitory to it. But there are other issues that strep can cause, and oftentimes antibiotics are warranted to get rid of it, to prevent other issues, truly, and really get rid of it. And most of us aren't going to gargle 17 times with salt water in a single day, and we're going to do it for 5 days. You're better off getting some antibiotics, and after your first day of antibiotics, you're already feeling better. You better keep taking them, but you're already feeling better, all right? All right, here we've got some bacillus. These guys, the gram-positives, Bacillus cereus, and then the pink ones that are a lot smaller, this is E. coli, Escherichia coli is E. coli. You know it is E. coli, right? You've probably never heard of Escherichia, right? But that's him, right? And I think Escher was the guy that identified E. coli in the first place, named after him. Here's another image. This is a blood agar plate. You can see it a little bit better, and the bacillus growing on it, not E. coli, but the bacillus, and these guys growing on there. There are lots of the bacillus. Really, if you give them nutrients like whatever, you know, the blood agar in this case, but nutrients to grow on, they take off. We call them, you know, a swarmer. They just grow, right? If you have a situation where you've got E. coli and bacillus together and you try to plate them out, you're probably going to be super hard pressed to find any E. coli as visible, distinct colonies on that plate. You're just going to see boatloads of bacillus because they completely overtake it and grow over the top of E. coli. E. coli might be there, but bacillus grew over the top of it. All right, it's like, whatever, I don't even care you're here, right? I'm going to feed on this blood, right? And they can partially hemolyze. They're weakly beta hemolytic, a little more alpha. Alpha hemolysis is the partial breakdown of it. You can see some that are a little bit beta, right? Here's some staph aureus with the clusters, right? We looked at those guys before, and you'll definitely see staph in the lab. A couple of different types of staphylococcus, and we'll show you some techniques to look at them and differentiate. Staph aureus is a common player on our skin, all right? We've all got staph aureus, all right? Some of us potentially have MRSA, all right? Some of us maybe have gone to the doctor for MRSA, all right? But some of us may have MRSA on our skin and we don't have any issues with it because it's still on our skin, all right? But if you get a wound, if you get a cut or a scrape where that MRSA bacteria was, and now all of a sudden you've opened up this area and given it opportunity to get into your body, now you've got MRSA, all right? And MRSA is methicillin-resistant Staphylococcus aureus, all right? That's MRSA, all right? Common, all right? Wasn't years back, but now it's really pretty common, all right? And with that, you may get treatment if you actually get an infection, a staph infection, you get treatment, but what you're treating is the bacterium inside of you. It's not treating the bacteria that's living on your skin, all right? You don't really get rid of the MRSA, all right? There are individuals that have had MRSA infections and have been hospitalized repeatedly, all right, because of reinfections, all right? So it depends, right? And it warrants the utilization of other antibiotics like vancomycin. Vancomycin is, you know, one of the big antibiotics that is kind of used in last resort for issues. And now we transition from talking about MRSA and start thinking about VRSA, the vancomycin resistant Staph aureus, because resistance happens. And if you start treating with all sorts of antibiotics, you better assume there's going to be some potential issues, right? And that sets the stage for what do we do now, right? VRSA was what we had, right? There's technically another antibiotic that we can step up to, but they try to hold that on reserve, right? And deep down, you know, if you don't need to be taking antibiotics, you sure shouldn't be, right? All right, this, oh man, this looks like a cauliflower to me when I see this, maybe with a little butter on the top so it tastes good. This is Mycobacterium tuberculosis. It's grown on a special type of media called Lowenstein Jensen agar. And we can see these cauliflower-like colonies. They're distinct characteristic of tuberculosis, all right? We showed some staining stuff. We showed the acid-fast staining, and I mentioned acid-fast staining was something like this. Once we see that acid-fast bacilli, it's done deal. We know you've got Mycobacterium, and we start treating with antibiotics. We're still going to take that sample and cultivate it. We need to grow it up. We need to grow it up into quantities that allows us to test it against a battery of antibiotics, because it's likely that the drug we've prescribed, we hope it's effective, but it's likely that this bacterium is resistant to that. And so you need to know, is it resistant? If it is, will this other drug be effective? All right. We treat because growing it takes a super long time. You can grow bacteria like E. coli up, you can inoculate it today and you can see it tomorrow, all right? But you could take a sputum specimen from a person that's got TB, we know they do, we saw it through our acid-fast staining, but you put it on the media and you look at it tomorrow and you see nothing. You look at it next week and you see nothing. It could be 2 or 3 weeks, maybe even a month down the road before you actually have culture confirmation and have enough organism to actually do susceptibility profiles, all right? Kind of crazy, all right? And part of it's like the mycolic acid. It takes a lot of energy, takes a lot of time to be able to get all of that going. It, you know, for them to replicate from 1 cell to 2 takes a super long time, all right? We've seen this before, this tree of life. And again, we're focusing on these two in this chapter, all right, as we think about the prokaryotes, all right? Here, we're looking at a deeply branching bacteria, Acetothermus. And what we mean by that, this guy is ancient, right? Deeply branching, it's split off very, very long back. Luca down here is the last universal common ancestor, right? That's Luca, right? And this in turn, and we think, you know, it's not just one organism that gives rise to everything. There were handfuls of single-celled microbes, microbe-like, bacteria-like organisms that were these first types of cells, and in turn, all sorts of interactions between some of them. This universal common ancestor giving rise to the diversity that we see here, but really not just one cell, right? Handfuls that in turn, now we get this last common ancestor. So the Acetothermus, deeply branching because it is super ancient, right? As far, you know, and as we look at this, we can get some time frames with, you know, this being, you know, billion years old, right? Maybe even older, right? We can go back as we're thinking about some early ancestral precursors to what we see, tracing life back. You know, we say our planet is 4.6 billion years old, give or take a couple of years, right? 4.6 billion years. And we can recognize evidence of microbes from almost 4 billion years ago, all right? And we keep pushing that back a little bit farther, all right? Almost 4 billion years ago, there were bacteria-like cells, all right? And this one is not much older, right? It's been around a super long time since, you know, and as we see this, we see a branching at this node right here, a split between the bacteria and the archaea. This node, a split between the archaea and the eukaryotes, right? But this, very ancient, right? Pretty impressive. This one's just flat out mind-boggling. Deinococcus radiodurans, they call him the Conan the bacterium. How do you kill it, right? It survives in the harshest conditions on Earth, for sure, all right? It's capable of surviving, thriving, really, in nuclear reactors, in the cooling ponds for the nuclear fuel rods that you simply touching one of those, Homer Simpson with it in it, you know, the back as he's walking out of there, right? You're dead, right? Deinococcus is like, sweet, right? That's what I wanted, right? I eat radiation for dinner, right? I'll eat your plutonium, right? The Department of Defense, the Department of Energy, both of these departments that are really pretty related, even though they seem like they're not, they are. These agencies are super curious about this type of stuff, right? What's up with it? And then we also start questioning things like, you know, astrobiology. We start thinking about, is there life out there, right? Something like Deinococcus that can survive super high levels of radiation. Don't you think that makes sense that something like that could be a microbe that could thrive in some other locations? Highly likely, right? So they're interested in things like that for maybe extraterrestrial life, right? Doesn't have to be a little green Martian, right? Extraterrestrial life could highly likely just be single cell microbes, right? Crazy, weird things, right? Not necessarily green men, right? And you know they're green because they're full of chloroplasts and they're sitting there photosynthesizing by themselves. It all makes sense, right? That's a completely different story, right? So these super impressive, right? And the ability to tolerate and protect themselves. They can survive, I can't even remember the levels, 100 or 1,000 times the level, it's got to be more than 100, it's upwards of 1,000 times the level of radiation that would kill you, right? And it does nothing to them, right? Very impressive. There are other instances. Our halobacterium, it's not surviving high doses of radiation, but it's something that's also pretty highly resistant. There was a researcher that was here a handful of years back, and they were studying kind of same idea, like resistance and radiation resistance, but using halobacter instead of Deinococcus. And they had some funding from the DOD and DOE to look at some of that stuff, right? Very odd to be in the biology building, walking around, and then you turn the corner and here are all of these military generals or whoever they are in, you know, their dress, and you're like, what, right? What's going on, right? X-Files type of stuff, right? You're like, what's up, right? Another one, as I see your Bigfoot shirt, walking in the building, and here's this little train track right down a hallway. And I'm like, what's going on here? And it was Discovery Channel filming Dr. Meldrum, who is a big Bigfoot researcher, right? That was completely different than the military guys. But another one that completely caught me off guard is I'm like, what? What's going on? I'm like, do my hair up, right? What's happening here, right? It's out there. All right, sulfobius, this is an archaea, and it oxidizes sulfur, stores citric acid in its granules. That's pretty impressive, I think. And we can maybe see some vesicles coming off of there. I'm not exactly sure what that is. Another electron micrograph as we're looking at it. The archaea, as we are starting to transition a little bit into this, They were classified as extreme bacteria before they got bumped into their own domain. Bacteria that could live in places like I was describing at the start of this lecture, where you think there's just no chance they could be there, right? The archaea potentially are, right? And this, where we've got these crazy bacteria that are full of sulfuric acid, you're like, how could that be, right? There's all sorts of potentials with some of these crazy microbes that sets the stage to give them a leg up in these environments. So as it oxidizes this and creates that, it sets up the situation for it to survive and harbor some of this. I'd like to say that this is Salt Lake, but it's not. But similar thing, and this, I did see something almost exactly like this as we flew over the lake the other day. A bunch of areas where the lake has been walled off, the water within there is allowed to evaporate, and what's left behind is salt, right? And here we've got some various bits. We've got more white. Here's some of the halobacter with the pinkish colors, and there's a handful of them as we're looking at it, and other stuff. This might be more liquid. We may have more water within here, right? And then they'll go in and dig it up and deal with the salt, right? But the halobacter growing within here, the pink sea salt, right? And if we're talking Salt Lake, we're not calling it Himalayan, right? But there you go, all right? Oh, and there's my exercise. I know I'm at the end. That's perfect, because I know you guys are probably done, right? We still have a few minutes, but this is right where I wanted to start, or stop, not start. Okay, this, I'm not talking about any of this. This is just showing, I believe, probably some filter and that bacterium on here, flagellate, and a couple of others. Questions or concerns? The whirlwind. All right, we will, oh, stop for me. Okay, we will do the next two chapters. We'll talk about some eukaryotes and then the virus. The virus chapter is a little bit less, although there's still a lot of stuff in there. So there shouldn't be any issues of covering both of those tomorrow. Good? Okay, guys, have a great lab. Have a good lunch. Hopefully you eat something before lab. It's a full day of microbiology. Good times. All right, see you guys tomorrow.